Thanks, everyone. I'm really happy to be here. I wanted to thank the laboratory and John specifically for inviting me, and Miles, who has been constant with me on email and making sure everything's set up, and of course, Simon, who is stellar at IT. I really appreciate being here. I'm kind of wondering why I'm not here more often. I already talked to Anya about maybe coming here every once in a while and using an office because it's beautiful and about 40 degrees cooler than Davis right now. So I really appreciate it. Okay, let's Simon, we're frozen, I think. Just about to get started. Let's start. We'll get there. Yeah, no worries. Give it a shot. Okay, great. Yeah. So this is an outline that we're gonna give you, and I'll kind of bounce through it because I'm gonna cover, there's lots and lots of things I get to work on and I have lots of fun doing, but I don't have time to cover everything. So I think we'll be about 45 minutes. I'll give you a little bit of introduction, a little bit about myself, what I do, some of the projects. We'll then talk specifically about some of the research topics that I'm involved with, most notably Klamath Dam removal and salmon life history, primarily Chinook salmon. We'll talk about beaver facilitated wetlands or off-channel habitats, which we have a lot of research going on with that right now. And then spring-fed rivers, some positive species interactions, and then how food webs might inform resiliency and growth for wild fish. And then I'll end with some of the more recent work that we're done. I'm excited. I actually got really excited last night because I was looking at some of the figures my postdoc pulled, and I'll get to show some of those for you guys today as well. We were on the phone late talking about them, so it's very fresh. Okay, so I grew up on the East Coast. I've been in California for about 25 years. I love the weather. I think that's why with my East Coast upbringing. I'm a systems ecologist. I look very broadly at ecosystems. So I did my graduate work with Dr. Peter Moyle at UC Davis. I was kind of always like, not a pure fish biologist. I operate at multiple trophic levels. I think that's how we describe it, right? So I look at a lot of food web dynamics and how that influences fish, but I also do a lot of fluvial geomorphology, hydrology, physical environment stuff as well. I was trained actually as an invertebrate taxonomist. I worked at the UC Santa Barbara Sierra Nevada Aquatic Research Lab with Dr. Dave Herbst for about 2 years. That's when I really found that I wanted to be a scientist and work on this, and that's kind of how I started, and then got more into fish ecology with Peter Moyle, of course, at UC Davis. I take a whole ecosystem approach. I'm extremely interdisciplinary and collaborative. Miles mentioned I'm associate director at the Center for Watershed Sciences. Prior to that, I was a researcher at the Center for Watershed Sciences. So I get to work with economists and lawyers and hydrologists, engineers, and of course, ecologists as well. It's a fantastic place. And I love working there, and my department as well is highly interdisciplinary as well. I bring a very diverse tool set to the table. Some of these tools I'll get to talk about today. Manipulation experiments, microchemistry of fish ear bones, etc. This is kind of where it started for me. So this is Blue Creek on the Klamath. I was young. I was a junior in high school. My parents took me across the country in a 1989 Suburban with my 3 sisters. We drove all the way out to look at colleges, and by the time we got to around Colorado, but really California, I just fell completely in love with rivers. I grew up in the Connecticut River Valley on the East Coast, but this was different than anything I had ever seen. And that was kind of my hook from the beginning. I started becoming really interested in salmon and started thinking about salmon more. And I realized what a phenomenal complex migratory life history these animals have, and I became hooked again and contacted Peter Moyle to work with him. This is Eosomo driftwood densis, which is the oldest artifact of salmon found in British Columbia about 50 million years ago, which is very cool. And I began to wonder, how could this fish be around for 50 million years? How is that possible? And so I started looking into their life histories and was just absolutely blown away. The phenomenal thing is this animal has weathered mega droughts lasting greater than 100 years, several hundred years. They have gone through tectonic uplifts and land change patterns. river disconnections, volcanic eruptions, and of course glaciation. And so I have a little bit of history there too. And I was like, how is this fish still around? And the reason it is because it has this immense and diverse life history, right? That allows it to, while some populations may become extirpated due to climatic events or anthropogenic events, it can come back because it is very good at straying, moving into new habitats, and it's very diverse, right? We have a diversity of life history traits with these fish, and that is bode well for it up until very recently. I quickly, when I moved to California, realized that Blue Creek on the Klamath or the Smith River north of the Klamath was not the norm in California. I realized that the vast majority of rivers in California have been grossly transformed through agriculture and diversions and levees, cutting off habitats that were critically important to their life cycle, such as floodplain habitat, dams on Matilaha, the Klamath River, that have blocked historical spawning and rearing habitat for these fishes. Hatcheries, production hatcheries specifically, not conservation hatcheries, which have put a lot of genetically homogenized fish into the system. And there have been a host of reasons why that is bad for wild fish and why we see a reduction in life history in wild fish and general land use patterns as well. This is up top there is one of my study sites, Big Spring Creek on the Shasta River, which is a cold spring-fed river, which I'll talk about in a minute here. But you can notice that the land use practices here needed some updating. We were able to help landowners with that. We have flood irrigation going on. That water is moved over the landscape about an inch deep. It heats up to 90 degrees Fahrenheit and that comes back into the river. And that's very bad for cold water fish that need very cold water, 15 to 20 degree water. You can also see cattle, those black dots all around, actually in the river grazing aquatic macrophytes, which I'll talk about extensively a little bit later. I think changing California river hydrographs might be the best overall depiction of how quick the changes in the environment have created these disconnections between behavioral adaptations of these fish, life history adaptations of these fish, even physiological and morphological as well. So this is actually not a California river, it's the Green River in Utah, but I think the point is well situated. About 1960, we have a natural flow regime here. On the x-axis, we have time and year. On the z-axis, let's call it the y-axis here, we have date, month, and our last axis, we have discharge, right? And what we see is historically what have happened here is we have very dry periods between October and April, and then we have very, very wet periods between April and September. A dam went in, Glen Canyon Dam, just about after 1960, and we see a drastic change in the flow regimes of those rivers. And for fish and for any native species truly that's adapted to the system, that's a problem because they have evolved to this over millions of years, and we change it within a week by closing a dam, and all their cues that cue their life history, cue their behavior, cue their physiology, they make no sense to them anymore, right? They lose their competitive advantage. That's why we see a lot of non-native species move in, etc. I think another way to think about life history diversity and why it's important, I think in the context of salmon, is habitat. If you could, please think of it as a life history diversity kind of incubator, right? If we have more diversity of habitats across the landscape, we tend to see better diversity in the life history of animals, whether it's fish, mammals, semi-aquatic mammals, aquatic insects, or plants. Life history diversity ultimately leads to population diversity, and population diversity across the landscape leads to species resilience to change. The problem in California and really throughout the West is that if we have these populations across, let me go back one second here. If we have these populations, we have a life history trait on our x-axis and frequency of that trait on the y-axis. And so we could use something like life history timing of juvenile salmon moving out to the ocean. If they move out to the ocean at different times, I was just talking about this with Lucas, there's a great bet that overall the species complex will do quite well and be able to weather disturbance events, right? If we don't, and all the fish are doing exactly the same thing, then we don't have that at all. We do not have a diverse set of populations, and what's going to happen to one population will likely happen to all populations. This is heavily influenced by Daniel Schindler's work that came out about 2010. And so I like to think of this outmigration timing to the ocean. Some fish might get there early, they miss an upwelling event, they die. But you have diverse populations. So some fish are going to show up later, and when they get to the ocean, they receive the upwelling, they can actually grow, they can do well, they can return as adults. The problem in California is we have put dams and most of our major river systems, over 1,800 dams in California alone. And those have reduced some of that life history diversity throughout the state and throughout the Pacific Northwest. We've changed our Central Valley rivers in particular, but really rivers all throughout the West. And in doing so, have eliminated important wetland habitats, floodplain habitats, et cetera. And that too has reduced life history diversity across the landscape. And finally, when we introduce hatcheries, the production hatcheries, the genetic effects, the behavioral effects, we've lost more. And so what we're left with is this kind of homogenized population exhibiting little growth or life history diversity and an inability to adapt to current conditions. So one of the main questions for me, let me just zoom this in a little bit. One of the main questions for me is, how do we get back to something like this? These fish are the same age, they are from adjacent rivers, they're the same species, but they look very different. And so I began to ask questions, where is this mechanistically? What's happening here? How are these fish different? How is one larger in size than the other? Why is one smaller than the other? How do we identify these productive and diverse habitats and determine the different factors affecting life history diversity within these habitats? And really getting back to something like this. So that will kind of move into the research. I have three main projects that I wanted to talk to. The first one being the Klamath Dam removal in wetlands and also spring-fed habitat in the upper basin. We know historically the Klamath Basin was the third largest salmon producing river in the Pacific Northwest on the West Coast, behind only the Columbia and the Sacramento River. Part of the reason for that was because of the diversity habitats. We have Upper Klamath Lake with lake wetland habitat. We also have adjacent off-channel habitat along the Klamath River. And then we also have major spring-fed rivers, which are rare river types in the upper basin, rivers like the Williamson, the Sprague, and the Wood River as well. Yeah. And we know that what we saw was production of 3 runs of salmon and also steelhead and also coastal cutthroat as well. And these fish were really important for commercial fisheries as well, but arguably not more important than Native American culture. These aren't actually salmon, these are actually endangered sucker populations in the upper basin from the Klamath tribe. And so sustenance fishing was really important too. These fish since this time, which is about 1957, have been listed and endangered, both species, and are really run through a conservation hatchery. And I get to work with some of those fish and the Klamath tribes, which has been a lot of fun. The big deal in the Klamath was in 1961 when Iron Gate Dam went in. Prior to that, Copco Dam went in, and we had J.C. Boyle and Copco 2 as well in that system. So 4 big dams. And I can express to you from like an ecological perspective the effect that these dams have had on fish. We used to have 100,000 spring run in this system pre-dams. There are less than 1,000 left, essentially regulated to one river, the Salmon River in the lower Klamath system, right? So that should give you an idea of the effects of these dams. We've had massive fish kills. In 2002, we lost 30,000 Chinook salmon because of low oxygen, because of disease, bacteria, et cetera, in the system, largely associated with dam and the movement of water through the reservoir systems. Okay, I think I'm going to mute this real quick for Zoom. Sorry, Zoom. and hang out with Barry for a couple days and raft the river. And most importantly, I want to say that all the research that I'm going to talk about a little bit today in the Klamath, is in direct collaboration with the Yurok tribe and Barry's group here.
You're gonna have to fast forward to the slide.
Yep. Okay, bear with me, you guys. They do that? Simon. Hang on. I can click on it, but it wasn't working for the.
It should work now.
Okay, so one of the main questions that Barry and I had was, how can we quantify changes in life history diversity associated with the largest dam removal in the world? So the removal of 4 dams, I'm happy to say, is done now. We used to say it's coming, but it's actually done. The 4 dams have been removed, and we're beginning to get our first fish back above Iron Gate Dam, actually moving into Oregon. Most of their heads are in my freezer in the lab, which is something I'll talk about in a minute here. But we are really interested in life history diversity, and I found it very timely that we were able to, I was just like, this is amazing. We're going to get to study this, right? Beyond the cultural significance of this and economics, certainly for commercial fishermen, the science we felt would be really interesting. And so we are actually interested in kind of the movement and the recolonization process of fish into the upper basin. And to do that, we're using fish ear bones. So we're using otoliths from salmon. So annually, we work with U.S. Fish and Wildlife Service, and we work with the Yurok tribes. And fall run Chinook are coming back to the Klamath River, and they're spawning and they're dying. And at that point, the Yurok tribe and Fish and Wildlife Service run surveys, spawning surveys, and they remove the otoliths, these ear bones, which are in the head. They have 2 of them, and they send them to us, to our lab. We're working with Rachel Johnson from the National Marine Fisheries Service as well, and the Center for Watershed Sciences at UC Davis. And we can get a lot of information from these otoliths that you see here on the right. So the standard stuff that we've done historically is looking like how fast these animals grow. We can back calculate and figure out when mom spawned, and particularly backdate their age and know exactly how old they are, particularly if we have thermal regimes, so we know that, and how long their incubation process was, and when they began exogenously feeding on insects in the rivers. That's kind of historically how we've used otoliths, and we've published some work on that. But this one was a little bit more innovative and really just came out around 2017 to look at the microchemistry of the otoliths. And to do that, we had to do 2 things. One, we needed otoliths from adults annually, and we were able to collect 4 years of otoliths from 4 separate cohorts of Chinook salmon returning to Klamath drainage. And then 2, we had to go around the basin and sample water. And we were sampling water for strontium, a strontium signature, if you will, which is just a naturally occurring element in the water. We took about probably close to 200 water samples throughout the basin. So any different tributary above and below a confluence, the main stem Klamath, we would take samples. And what we were looking for is there a good diversification of strontium in the water based on the river, right? So if we went to the Wood River and sampled it and took a water sample, what is that strontium signal? Versus maybe Blue Creek, which is much further down. Once we have what we call the isoscape set up, which is just the signatures of these individual tributaries and main stems throughout the basin, we can then look at the otoliths and using a laser and then ablate the otolith and capture essentially the dust that we're ablating and pull the strontium content from that and then match it up in the watershed, right, based on its age. And so it's like the otolith is this kind of like little geolocator in this fish that will tell us the life history of these fish. The cool part again is, if you remember, I said these are adults coming back and we're taking their otoliths. We are going back in time when they were juveniles during their first 4 to 6 months in the river, and we can isolate that. So this is the isoscape. Francisco Bolito Leyva is my postdoc. He's working very hard with me on this. And we were able to define the isoscape. And where you see the different colors suggests that we have difference in strontium values. And if it was all one color, you would say, oh man, it's going to be hard to tell what's going on. But as you can see, we have a lot of different colors going on here, which suggests there's a diversity of strontium values in this watershed. The strontium varies with geology. The Klamath is a very active location. There's sedimentary rock, there's metamorphic rock, there's volcanic rock, and we expect that those rocks truly influence the strontium content. Kimberly Evans also helped a lot on this as well. Not in a little bit. She's now a grad student at UC Davis and just wonderful. We can then look at the strontium isoscape in the Klamath watershed, and we can look at the life history profiles that you see here on the right. So on the x-axis, we have distance in otolith, core to edge. And on the y-axis, we have the ratio of strontium 87 to 86. That blue dotted line across the top, that's the ocean. The strontium value in the ocean is very standard because of all the mixing and whatnot in inputs. It does not change very much at all. We can't tell very much about what their life history looks like in the ocean, but what we can tell is what it looks like during the freshwater residency period you can see here. So the otolith core is heavily influenced by the mom because the otolith core, the juvenile at that point is an embryo and is feeding on maternal yolk, right? But as soon as they hatch and they move out, they begin to incorporate the strontium signals of that river into their ear bones. And so what you see here, for example, is 1, 2, 3, 4 different fish. And basically, it's not this simple, but you just match the colors, and then you can tell where these fish were born. We can also, based on size classing, understand, and because we see when they reach the ocean, we can understand their marine entry date. We can also understand how large they were. The important caveat here is we're only measuring the winners, not the losers, right? We're only looking at the otoliths that we actually get back from the adults. The ones that don't make it back, we don't know. So we really wanted to ask a question, how does dam removal affect life history diversity? I wish I had more data, but we just have those otoliths in our freezer in the lab, and so we're just slowly getting into them. This is pre-data, and we've identified probably 12 to 15 different life histories pre-dam removal, and we expect that when the habitats open into the larger basin, fish can access those productive spring-fed rivers, those wetland habitats, those off-channel habitats, that will see significant benefits in life history diversity in these animals. Okay, the second thing I want to talk about is kind of beaver wetlands or off-channel habitat. So do beaver wetlands promote aquatic biodiversity and growth diversity of juvenile fishes? So again, remember, we're talking about, can we find diverse habitats? Where can we get the greatest bang for our buck to promote resilience in fish? And so this is another project I got to work on with numerous grad students, which I'll thank at the end. You're also seeing their pictures, if I didn't mention that earlier, that have been working with me on this. Okay, so how do beavers and their engineered wetlands affect biodiversity and aquatic species? There have been precipitous declines of beaver throughout the West for since about 1859, if you can imagine. They enhance, we know from Michael Pollock's work, they enhance habitat heterogeneity and complexity. They enhance water table and provide cold water during summer, typically warmer water during winter. So in really cold places in Northern California and Southern Oregon that I work in, warmer water in the winter is beneficial to growth and survival. And so how do beaver and their habitats promote biodiversity and the growth diversity of juvenile fish? I'm going to talk a little bit about insects quickly. This is a paper written by my postdoc, Nick Corline at Virginia Tech. He's at UC Davis. He finished his doctorate at Virginia Tech. And we had this unique situation working with the Scott River Watershed Council where we had two primary habitats. We had main stem French Creek and we had a side channel on French Creek. And Scott River said, we want to study what the effect of if we put BDAs in, beaver dam analogs, into the system. If we build them, can we capture the environmental diversity that they will provide across the landscape, but also to the fish themselves, and can we capture that? And of course, as any good researcher, yes, we can do that, definitely. And so we decided to use aquatic insects as kind of our end game, or our model here. And so what you're seeing here is in 2017, only 2 habitats existed. So we have side channel habitat in yellow, and we have the blue greenish habitat is the main stem French Creek. And we looked at aquatic invertebrates. These are quantitative Venn diagrams. And you can see that there were 23 species alone in the side channel habitat that were not in river habitat, and 16 species in river habitat that were not in side channel habitat, and 34 species that were shared between. We had a gamma diversity or a regional diversity across the landscape of 73 species. But we were really interested in what happens when these BDAs go in. In the first year of the BDA implementation, we saw a real boon in the insect production, and we saw a real bump in the diversity as well. So immediately, we saw numerous species, 13 occupying. These are new habitats. We expect those habitats to take some time, even with insects, to colonize, develop, and reproduce. But almost immediately, within 2 months, this is the data we saw. We saw a large bump in the biodiversity of aquatic insects, the gamma diversity, to 86 species overall. 1 year later, that habitat matured a bit more, and we saw a large increase in gamma diversity throughout, suggesting that if you build the habitats that these animals need, that we will see an equivalent response to biodiversity. And we actually saw that with the insects. It's part of a second project that we're actually working on now, and it's too early to show you guys data, but I wanted to give you some preliminary data. We're also interested in what are the effects on fish. And so we looked at 3 primary metrics. We wanted to think about macroinvertebrate density here on the left. So we have time on the x-axis and density of insects on the y-axis. Macroinvertebrate density is very important for fish because juvenile fish rear and they feed on aquatic insects, right? So the more food you have, the better it generally is for fish. Streams are naturally oligotrophic or unproductive, right? Most freshwater streams, not all. Some of these side channel habitats, as we see, we're producing quite substantially much higher food resources for rearing juvenile coho salmon. We also looked at temperature, and we have the river here, and we have the wetland habitat, and we have, again, time on the X and mean daily temperatures on the Y. And what you can see is we have slightly warmer water temperatures in our BDA, or beaver dam analog habitats, 5 degrees Celsius. And in the river, we're getting very, very cold. We have ice up conditions and whatnot. We typically see a lot of high mortality there. And then in the summer, in those habitats, these BDAs essentially provide a pool habitat off channel, which is a little bit cooler than you would expect in the river itself. So again, habitat heterogeneity to provide biodiversity and possibly life history diversity for fish. This is some very kind of anecdotal, it's not totally anecdotal, but low numbers, but we're beginning to see more of this in the data we're looking at now. We're actually studying 4 different river systems with BDAs and rivers. But this is based on marker capture effort for coho salmon, looking at mass growth rate for coho salmon, and fish that were caught in wetland habitats exhibited significantly higher growth rates than they did 10 feet to the right in the adjacent stream in a pool habitat, which was very neat as well. Okay, 1 sec. I want to take a sip of water. Okay. A lot of my work as a graduate student and to this day, Nick, my postdoc, is working on this with me, as well as Francisco, is looking at spring-fed rivers. I love spring-fed rivers. They are groundwater rivers. They occur in volcanic terrains, right? They are not precipitation-derived rivers. They're not influenced. They can be an influence a little, but not by snow, not by rain. They are large, anomalous springs that are showing up across the landscape and can deliver anything from 80 CFS to 150 CFS that we see in the Shasta River, which is a tributary of Klamath, to up to 900 CFS that we've very recently noted on McCloud River at Big Springs, which has been no small task to get discharge on those systems. And so I want to talk about that a little bit. Very specifically, I want to talk about the biotic mechanisms that might be responsible for improving life history diversity in spring-fed rivers. And then how do food webs affect growth diversity? This first question is really interesting because if you read the fish literature, most of it is about abiotic mechanisms affecting fish. What's the temperature? What's the discharge? What's the substrate? But coming from a food web background, I began to ask questions about what are the biotic effects? Where are we seeing species interactions? Is it possible that we see positive species interactions in these systems? we actually know quite a bit from previous work that I've done, right? So here we have temperature, and this is comparing runoff rivers to spring-fed rivers. So capturing this diversity and habitat, again, across the landscape. X-axis, we have time. Y-axis, we have temperature. This is our spring-fed river here. It's optimal. It doesn't change very much. It's very cold in the winter. Again, it's very warm in very cold in the summer and very warm in the winter, as opposed to a runoff stream where we see huge swings in temperature, 20 degrees plus in the summer in some of these systems, and very, very, very cold in the winter as well. We also noted that flow, this is a composite from a paper from Freshwater Science where we looked at 3 spring-fed rivers and 3 runoff rivers. You can see the large peaks in our runoff rivers. Those would be rainstorms or rain on snow events. You see a snowmelt recession coming down here to a baseflow period. We see on the spring-fed rivers essentially flat for the most part. We do have a little bit of runoff from Bear Creek coming in here, but very flat across the board when we compare. And then strangely, in June or July, we see this bump in stage in our rivers. And that's largely due to biotic conditions in the rivers. We see aquatic macrophytes growing and during the spring, and that's effectively like dropping ice cubes into a glass. Your stage goes up, right? And that provides more cold water habitat for fish as well. The last big one that I want to talk about is nutrients, and I'll talk about that extensively through the rest of the talk. So one of the important things is that we see naturally occurring nutrients. Again, naturally, this is not anthropogenic nutrients in spring-fed rivers. At these spring-head locations, we see 0.2 to 0.3. We've seen as high as 0.5 milligrams per liter in places like Big Springs Creek and the Klamath Drainage. That is, we propose, nitrogen coming from ancient marine sediments from when California was an inland sea. We've had a paper in review for a long time. It's not quite out yet, but we're getting there. Whereas in our snowmelt system, we have very low nutrients, okay? And just think about nutrients as almost like fertilizer, which it is, right? And so it's very typical when we have high nutrients, we have high aquatic plant biomass in this system. You need nutrients, but you also need a flat flow regime too. If you have big spikes in your flow, it will rip the plants out. And so one big difference is thinking about the physical habitat. Has anyone ever seen a river kind of look like this? A little bit, maybe? The chutes or something like this? It's very rare. These are rare rivers in the landscape, and they look different. So we began to think about, what do these plants mean for fish? At the time, I was working with my colleague, Carson Jeffries, at the Center for Watershed Sciences. He was doing dive surveys out there, and I was collecting a lot of the food web work. And we recognized that when he looked at steelhead, they seemed to preferentially select aquatic macrophyte habitat in these spring-fed rivers. So when we compare these different habitat types, backwater cutbanks, emergent beds, large woody debris, point bars, the dominant place where we saw juvenile steelhead rearing was immediately behind or actually within aquatic macrophytes, as I just showed you. And so we started to think about how might the submerged vegetation positively affect the growth and our life history diversity of native fishes. They must be choosing this habitat for some reason. So what is it about this habitat? And we started thinking about some of Jay Stackowitz's work with positive species interactions and Hutchins's realized niche and fundamental niche, right? And so the fundamental niche, I don't need to explain to you guys, meaning the full range, without predation, competition, etc. The realized being with predation, recruitment limitation, disease, parasitism, etc. But if you have positive species interaction, that might be able to push a realized niche past your fundamental niche. And so that's kind of the theory that we were thinking about a little bit with this. So we decided to run an experiment, habitat manipulation experiment, really just looking at the aquatic insects to start, and then we moved on to the fish. And what we did is we set up these paired plots, and this is no small work effort, right? So like choosing the plots, we actually choose 2 aquatic plant plots. We fully remove one. We pull all the gravel out of the river. We let it sit on the side of the river to 2 months, dry out. And then we put it back into the river for a month so it incubates, right? And then we are really interested in kind of 3 things if you're a fish. We thought about food. We thought about drift, so benthic biomass, so how many insects are in the plants versus the gravel habitat. We thought about drift, so that's when aquatic insects move off these habitats. That's when fish are feeding. If you ever see a trout feed, it's sitting right there picking off drift. And then we thought about velocity as well as possibly bioenergetically, maybe this habitat is favorable for fish. And we found some really interesting results. So on the top, we have velocity. So don't worry about the dates. We have gravel habitat and submerged habitat, a submerged vegetation habitat at 20% depth, 60% depth, and 80%. Fish generally feed at about 60% depth. They've been known to feed at both 20 and 80% too. We wanted to see kind of what it looked like. And what you see is behind the submerged vegetation, at 20% depth, velocity was lower, right? So these plants are almost creating some sort of an eddy effect that might be beneficial to fish. At 60% that grew, to some extent in May when we had high biomass, we're actually seeing negative velocities. That is an eddy. That means the water is moving upstream, right? We also saw that later at 80% depth in almost all the habitats as well. At the same time, we were comparing aquatic invertebrate production over 3 periods, and we noted that the macroinvertebrate density, or the available food or resources these fish, was around 40,000 aquatic invertebrates and then decreasing into May of 2009, whereas the submerged vegetation had this kind of third dimension of habitat, and we saw extremely high values. These are some of the highest benthic macroinvertebrate data, invertebrate density data that I've seen, and I've sampled hundreds of rivers in all of California and Oregon and Washington. We saw extremely high densities, 2.5 to 3 times the density of food in these systems. And then we ran a drift study here on the right, and we just put drift nets. These are nets that are just sitting in the water. We measure it in the hour before sunset and the hour before sunrise. And we had those nets behind our plots, only gravel or only plants. And we saw that the drift density, so those are the aquatic insects that are drifting through the water column that fish feed on, were 2.5 times greater in the submerged vegetation than the gravel. And so I think the big thing that we took away from this is that there may be some positive species interactions going on here. I remember asking Jay Stackowitz about this when I was a grad student, because these fish are using that habitat. They're moving behind plants. They're experiencing lower velocity. They expend less energy, but they are getting tons of food while they sit there. Whereas if you're in gravel, you're expending more energy and you have less food. And that made a lot of sense to us. And we published a paper a number of years ago. Because of the food density in the systems, we became really interested in what it means on the effect on growth. CDFW had recently released a paper about temperature on the Shasta River, the very system that I've been studying since 2007, suggesting that for this species, coho salmon, I have my permits, I promise. For this species, coho salmon, they need water of 15.5 degrees Celsius. And that's by Steven Stenthouse. It came out several years ago. But at the same time in the literature, we noticed that Peter Bisson had put out some work that suggests he had seen feeding of coho at 25 degrees Celsius, much, much warmer water. And so we wondered how that might be possible. How could you have such warm water in a system and have these really cold water fish survive? How do we reconcile these results? And so the first experiment was hard. This one is even harder. We set up a manipulation experiment on the Shasta River and Big Springs Creek. We had a longitudinal experiment. So we start up top near the springs, very cold, low food, and we had a natural gradient. As we move downstream, water temperature would increase and production of invertebrates would increase as well. We set up 5 enclosures at each site, which are experimental replicates. We had 6 fish in each page. People laughed at me. They said, we'll never recover any fish. We left them out there for 60 days. I had nightmares about otters most nights. We did lose 2 cages due to otters. We were able to recover 125 out of the 150 fish. And we found some really cool results. This is a picture of the fish. So these were the fish in the coldest water temperature. It was a max temperature of 16.1 degrees Celsius. They showed a growth rate of 0.13 millimeters per day, or a percent change in mass of 26%. Downstream of the springs, where it was much warmer, we saw a growth rate of 0.27 millimeters a day, double, a percent change in mass of 161%, and a max temperature of nearly 22 degrees Celsius. And I think what we learned from this is that food is very important for fish. As I mentioned earlier, a lot of streams are oligotrophic. They're naturally They're not very productive. Spring-fed rivers are very different. And that's why I think they're so important, because they show a diversity of habitat. And so this has led to an aside, of course, that we're still thinking about, but can fish eat their way out of climate change? Probably not. But if there are slight increases in temperature, this is another reason why these rivers are so important. Because if there are slight increases in temperature, they can metabolically compensate for those increases by consuming more food. As their metabolism speeds up, if you're in a runoff river, you have to find colder water, you're going to starve, become sick, die, etc., experience mortality. If you're in a spring-fed river and water warms up due to climate change, even after other anthropogenic issues, you can have enough food to compensate, at least for slight increases. And we've been thinking about a lot of this as well. Okay, I wanted to kind of close with current research. This is kind of the big dream that I've been talking about in my department, but also with my postdocs, Nick and Fran, about the Pacific Rim of Fire and getting at this idea of spring-fed rivers, why they're different and why they're important for life history diversity for fish. So the big question that we're asking is, can geology be used to predict productivity and life history diversity globally? As I mentioned, we know a lot about these spring-fed systems. So these systems are integral to population diversity and species resilience. Paper came out just a couple of years ago. They show optimal thermal and flow regimes. They're extremely productive in terms of both benthic production of aquatic insects, but also algal production, periphyton production as well. But we're asking the question of why is that case? What are the mechanisms, again, promoting diversity and productivity at broad spatial scales? Can we capture those so that we understand it better so that can be applied in policy as well? You guys remember these fish I showed you earlier? It's like they're the same fish. They're the same age. They're the same species. They're from the same watershed. They're from just adjacent streams. There's one big difference. That fish on the left is from a basin that is metamorphic and sedimentary rock. That fish on the right is from a volcanic spring-fed system, okay? So again, the nutrients, the food, the optimal temperatures, the optimal flow regime all plays into this life history diversity of rainbow trout in this case. And I think we're going to see that in the Klamath as well. But we still don't know where the nitrate comes from. We have some hypotheses about it. The fertilizer for these systems is a paper we published in 2021 with a geomorphologist and Randy Dahlgren over in LAWR. And so typically what we think of is in runoff systems, we have snowpack pile up, let's say Mount Shasta. Let's not say Mount Shasta, let's say like the Sierras, right? That snowpack piles up, you have a lot of sedimentary, metamorphic rock, granite, etc., granite, and that will run off and become rivulets and eventually turn into rivers. In places like Mount Shasta, that's not the case. It's all volcanic rock. Have you guys ever looked at Shasta and been like, there are no rivers coming off Mount Shasta? That's because all the snow infiltrates through the volcanic rocks. It infiltrates the basalts and the andesites, and it moves down and it goes really deep in a lot of these springs. And we're proposing that it's actually going so deep that it's going through magmatic heating and that it's picking up nitrate, naturally occurring nitrate from crustaceans, etc., that are trapped in the sediment from when California was an inland sea, and then it's then pushing it out at these spring sites that appear anomalously across the landscape. That's kind of what we think. And so we actually got a grant recently to look at this a little bit, kind of expand on some of our earlier spring-fed river research. And we found so far, it's very preliminary, but I have some data to show you guys. These are 3 different river types. And again, we're chasing life history diversity here. And so this is fish weight on the, oops, sorry, you guys. This is fish weight on the y-axis and hydrology type on the, and geology on the x-axis. And we sampled 3 runoff streams, 3 hybrid streams. So that would be a stream that has both metamorphic and volcanic rock. So it has both a snowmelt pulse hydrograph, but it also has springs along it longitudinally, and then pure spring-fed systems. These are McCloud River below Springs, Rising River, Hat Creek, places like that up north. And these are the fish that we captured last fall that we're working on their otoliths right now. And I think the interesting thing to me. This is kind of what I propose, and it never really works. I feel like I have these hypotheses and thoughts, and then we collect the data. I was like, oh, that's not what I was thinking. But this one is actually what I was thinking, because we had an idea with those rainbow trout I showed you that those fish are larger in the spring-fed rivers for all those reasons I mentioned, and smaller in the runoff. And the hybrid kind of fall right in between. They're a little bit larger than these runoff rivers, and they're a little bit smaller than the spring-fed systems. We then have taken those otoliths and can do some back calculations. We're going to go back to curves. You guys remember I started with all those curves. So this is really nice. Nick plotted this up for me last night. And what you see is only 3 streams here, but this is estimated spawning date. So we see a very tightly controlled spawning date of rainbow trout in these runoff systems, which dictated by flow and temperature. But in these spring-fed systems, because it's so variable and there are fewer cues, we see a much broader curve in both Rising River and Fall River here, right? We see fish spawning throughout the year. And so we have fish of all different ages, and we have this immense diversity and in life history of these fish as well. We can then look at the hatch date. So when they're actually hatching out of the gravels and beginning to exogenous feed, which is the next one. And we see something very similar. The hatch date for runoff streams is extremely tightly controlled by water temperature. So when fish hatch out of eggs, that they're incubating, right? So if you have very, very cold water, it warms up, you speed up, and then you hatch. Because we have such optimal temperature regimes in these spring-fed systems, we see hatch dates occurring throughout the year because we don't have, it's not super, super cold or super, super hot. It's just kind of just right. And then lastly, this is our exogenous feeding date. So that just means when the fish are not feeding on maternal yolk anymore, they're emerging, they begin to feed on aquatic insects. We see very similar data here as well. With our runoff systems, we have very tight controls on exogenous feeding. They hatch and they begin to feed in a very tight window. And we see much broader feeding strategies on exogenous feeding throughout the year, suggesting multiple cohorts in 1 year due to the multiple spawning as well. So these are kind of our big questions that we'd like to tackle. Can geology be used to predict productivity and life history diversity globally? We know disproportionately spring-fed systems support high diversity across the landscape. They act as climate refugia from temp, hydro, and food, which we've discussed. Spring systems are integral to population diversity and species resilience. At least we think so in that initial publication of the work we're doing now. And the early evidence suggests that hydrotypes, spring-fed, hybrid, runoff systems are important factors influencing life history diversity across the landscape. Yeah, and so these are my peeps. They all rock, and so I put them throughout. I try to. This is many of them. There are more. Not all of them are still here, but there are so many of my people, grad students, junior specialists, assistant specialists, postdocs in the lab that are just amazing. And none of this work is possible without them. I get to give talks and think about data, particularly with the postdocs, but they all work so hard. And the energy in my lab is amazing too. They just like care for each other, and I couldn't ask for a better group. So I want to make sure to give them a shout out. Thank you, you guys. Let's see.
Graphs you had black dots. Is that some special meaning?
No. It's like a smooth density curve. That's the last question I asked Nick last night. And so the height doesn't matter either. It's just like a smooth histogram that you can write in R. Yeah.
Okay.
Yeah. That was the last thing I asked last night because I was
Yeah.
wondering the same thing. Yeah.
Thank you.
Yeah. You guys have any questions on the Klamath? Yeah.
So for the Klamath Dam,
Yeah.
there must be obviously a huge change for the ecosystem. So I'm wondering, I guess I'm wondering if you think there's take some period of time for those habitats and life histories to like need some kind of new stable river, and that's something you're tracking over time. Like it seems like the initial change in life history might be different than what it's like 5 years after the dam or 10 years after the dam. So you have any plans to kind of look at how things change as that,
Through time.
and changes through time?
Yeah, so what I didn't get to talk about is we're doing a lot of, with partners, but also in our lab, looking at changes in the ecosystem through time with aquatic invertebrates in particular. So we have like 20 sites set up from Oregon all the way down to the mouth, that we have baseline data on. We have hydro data, we're working with sedimentologists as well, that kind of thing. And I completely agree with you. Rivers are wonky. They're never quite in equilibrium. It's always dynamic, right? And so I think we will see change through time, but I don't fully know. I think the interesting thing is this salmoniid life cycle for the animal we're looking at. I mean, we won't get, so the first fish came back and spawned in Oregon this year, and we won't see those fish for 3 years, right? So even the next 2 years, we're still going to be getting fish back from when the dams were still in. So I think it's actually will be neat to see that and then track it through time. And we actually just got a grant from Fish and Wildlife Service for 5 years to kind of look at that. So we're really excited for it, and that's a really good question. Yeah, thank you. Yeah.
The differences we're seeing between the spring fed rivers and the snow.
Yeah, like a runoff river. Yeah.
Runoff rivers. I'm not so familiar with standard stream, but how separate are the populations of fish though in these two river systems?
Yeah.
And are the differences, do you think the difference is that just some, you know, to the plasticity or like genetic differences between the?
Yeah. Yeah, I think there's yeah.
Maybe reiterate.
Yeah, of course.
For the Zoom people.
Yeah, so the question is there overlap between the populations and is there phenotypic plasticity between fishes? So my understanding is you're saying like if you're a runoff fish, you go into spring fed, they showed maybe a different phenotype, and then are they also overlapping in terms of their genetics? So for most of the fish I showed you, the answer is no, because they're above impassable dams. So the species of interest there is rainbow trout, and it's all in the Mount Shasta area and north in the Sacramento drainage above Shasta Reservoir. And they are mostly have barriers where they cannot intermingle. And so it's pretty distinctive. But on the Klamath River, we have the Shasta River, which is a spring-fed system. And most of the other systems are runoff systems. So 100% I think that. And we don't know if, I think we'll know, it'd actually be really interesting to look, like, because we have 4 years of pre-data, if some of those adults are born in a runoff river and choosing to stray maybe into a spring-fed river, and then what that might look like. That's a really good question. And of course, in the upper basin now, we have these 3 spring-fed rivers, the Williamson, the Wood, and the Sprague is partially spring-fed that they will have access to. They're not there yet in their first year. They didn't make it quite all the top, but they will eventually. And so I think there will be greater overlap there where we could see that. Yeah. Yeah. You guys are asking a question that makes me, I got to write this down. Yeah. Yeah. Yeah.
I was looking, so kind of you're mentioning a lot kind of about these geologic features of the river and the types of rock that they know that might be really influencing the fish life history. Do you know of any of the regions in the world that you might see similar patterns or have other people look at this maybe with different?
Yeah. So the question is I think, which I've gotten from my department too, is like, was this just a one-off for California or does this have broader implications? And so this figure is the one that I didn't talk about, but this is the Pacific Rim of Fire. So this is what you see in the background is volcanic terrains throughout Asia and then down south into South America as well. So we're working, I actually have a spring-fed scholar coming from Japan in the fall who works on these systems, and we're beginning to collaborate. So that's the big dream. We're starting right now with 4 spring-fed, mostly in Oregon, California, and we want to collect more of that data to see if this is a one-off or we're seeing this in other places as well. That's what we'd like to do, but it takes money, like big money, right? So yeah, very good question. That's the hope. Yeah. Yeah, Miles.
So there is a spring-fed river in my hometown, but most of the groundwater under the water table, they're slightly carbonated,
Yeah.
and the whole stream is also slightly carbonated. That's the case for
Other.
river.
We don't see, we do see carbonation, and I'm not a geologist, but we do see that in some springs, particularly on the upper Sacramento River. It's so good tasting. You can like just fill your bottle up on our hikes. But we don't see that in a lot of other systems for what it's worth. But water quality is not totally my expertise. So yeah. What river is it? In Southern California, Irvine or?
If I,
Oh, in China. Okay, okay. Yeah, so we see that a little bit, but not so much. It's kind of, again, anomalous in a couple locations. Yeah. Yeah.
and there's a question.
Yeah.
For the past million years, sea levels were much lower than they are now. Only twice, 125 Ka, 20 to 4 Ka sea level was higher for the same as it is now. Would any of this impact the fish, broader shelves, changing ocean productivity?
Ocean productivity? I met a great researcher today named Lucas that works with John that would be way better at answering that than I could answer that. I don't know. I think that it's funny. I was talking, is Lucas here? I was talking with him earlier about this, but we don't know a lot about the ocean. It's a big box that we don't talk about a lot. And you ever talk to a salmon biologist that works in the ocean? It's very, very rare. But if you consider the life cycle of these animals, 3/4 of the life is in the ocean, right? And so that's why I'm excited to work with John and maybe Lucas and thinking about upwelling and timing and life history diversity of these fish when they get to ocean, this mismatch of upwelling, when you get there, et cetera, right? I don't know specifically on that. John might have a better answer than I do as an oceanographer, yeah. Cool.
Wait, what?
Yeah, John.
And with that, why do they grow better in those systems? Like, you know,
Say again?
fish grow better in the beaver dams.
Yes.
Why do they do that?
Primarily, we think it's a bioenergetic thing, much like the plants. So out in a place like in Siskiyou County, Etna, Scott River, that's a watershed we've been studying for a number of years in multiple BDA locations. We see that warmer water in the winter, colder water in the summer. Those are very difficult for other fish in stream, that very warm water and then very cold water in the winter. And then we do see, and I didn't touch on it, I talked about the diversity insects, but we actually see much higher density of invertebrates in the BDA habitats as well. There's just more food for them. And there's very little flow. So it's like a pond habitat. They can grow large and then they can move out, right? So getting at those size class differences in these fish between these habitats, to me, that's what we want to see, differences in fish. That's not to say the biggest, most salmon biologists, the biggest fish, that's what we want, the big ones that get to the ocean. It's actually the diversity that we want to get to the ocean, right? The smaller fish may win out sometimes, particularly in like a drier year or something, or a wetter year, I should say, where bigger fish move out later and they don't quite make it, right? So that diversity is what we're looking for in these habitats. That's kind of what we're chasing. Yeah.
I had a dumb question.
No,
what's your
never.
fish?
What?
I have a favorite fish.
Do I have a favorite fish? I feel like my son asks me this all the time, and I thought about it and I had a good answer. I love salmon. I mean, coho are one of my favorites for sure. I love bull trout. They're not in California anymore. They were extirpated in about 1972. One of Peter Moyle's students caught the last one. He's very forlorn. There's a picture of it I show students as freshmen. Yeah, I just love salmonids. They're beautiful. They're amazing indicators of habitat. I love insects too, but I thought you were going to ask me what my favorite insect was. That would be Megaloptera, which is the beast out there that can do quite well, and it's a big time predator. So yeah, that's a great question. Yeah.
So with thing with big life and then spring that active as like habitat, egg habitat. And you're saying BDA almost mimics that same kind of like still water.
Yeah, like I just said. Yeah.
So there's increase of the diversity within, like, did you also know you know that the like plant life or plant experience?
Yeah.
Runoff would not.
Yeah, so we saw some plants, mostly emergent in the BDA habitat. We didn't quantitatively mark it, very anecdotal. But the big thing that we see, the big change that we do have quantitative data on is the organic matter input and the settling, right? So as a river is very transitory, it's moving, stuff's happening all the time. Sediment moves, organic matter moves. But in the BDA, we tend to see that organic matter from like coarse particulate organic matter from leaves and whatnot gets trapped. And that's just immense resource for aquatic invertebrates. And we think that that has a lot to do with why we see high densities of vertebrates, and then why we also see different types of insects in those habitats than we do in the river. Yeah,
It's time to wrap up.
you guys are best. Thank you.
And thank you, Rob.
Thank you, Miles. Yeah.